right, today we're going to be talking about nerve cells, or neurons, which are the cells that make up our nervous system. And in introduction, the human brain is made up of approximately 100 billion individual neurons. So you think about that, that's a lot of cells that make up your brain. And your nervous system has two kinds of cells. There are neurons that receive and transmit information. These are the cells that we spend the most time talking about because that's really the function of the nervous system, right? Is to send information from your brain out to your body and your muscles to do something, and then bring information in through our senses into our brain so that we perceive things, we see things, we hear things, and that's what the neurons do. There are also what we call glial cells that don't transmit information but perform support tasks for the neurons, like providing structure, protection. They make the neurons run more efficiently. And then during development, brain development, they guide neurons to the right place in the brain. The glial cells, interestingly, there are 10 times as many of them than there are neurons, but they're very small. They're about 1/10 the size, so they end up occupying about the same amount of space in the brain. Glial cells used to not get very much attention. They were just thought of as those support cells that kind of keep the neurons working properly, and the neurons were the shining stars of the nervous system. But now more and more researchers are focusing on glial cells, thinking that they may do some more important tasks than just support the neurons. And in psychology, we're interested in behavior and thoughts and emotions, and truly our behavior and thoughts and emotion depend on the communication between our neurons, which is why we need to learn about them. And it actually wasn't that long ago, if you think of this researcher, late 1800s, early 1900s, that it was first demonstrated that individual neurons were actually individual cells. They were separate. If you imagine looking at a brain, I don't know how many of you have seen a brain or a picture of a brain. I will show you one as the class goes on. It looks like one solid mass, one solid organ. And so it wasn't until, the late 1800s, early 1900s, where they used a staining technique called Golgi staining. And what happens when you use that staining technique is that there is, the stain ends up staining certain neurons perfectly. So you get these perfectly stained neurons and you can see those neurons exactly. And then the stain doesn't affect other neurons. And so by doing that, they were able to say, look at that. There are actually individual neurons, individual cells that make up the brain. And so that gave rise to the neuron doctrine, that neurons are independent, specialized functional units that are separated. And that's the key, because they're individual. They are not connected by a small space called a synapse or a synaptic gap, which we'll be talking about. And information has to be transmitted across the synapse from one neuron to the next. Look at that. A beautiful neuron with all of its, that we're going to learn about, the shape that a neuron is in. All right. So what is a neuron? That's what we're focusing on today. Well, like other cells in the body that you might remember, maybe from high school biology, neurons contain the following structures. And again, these are structures you would see in any cell in your body. There's a membrane that separates the inside of the cell from the outside of the cell. There's a nucleus that contains the chromosomes. There's a mitochondria that provides energy for the cell. There are ribosomes, which are sites at where new protein molecules are made. And there's the endoplasmic reticulum, which is a network of tubes that transport new proteins to other locations. And here's a picture, you can see here's the plasma membrane, or the membrane that's separating the inside of the cell and the outside of the cell. The nucleus that contains all the hereditary information. The endoplasmic reticulum here, it's kind of like a transportation system for proteins that are made in the cell and need to move to other areas of the cell, kind of like a freeway system. And then the mitochondria here that provide energy for the cell. The cell membrane is made up what we call a phospholipid bilayer. So bilayer meaning there are two layers, and phospholipid, lipid is fat. It's kind of like a fatty membrane, and there are two layers that's made up of these molecules. These, you can see each one here are these molecules that have what we call a hydrophilic head. That's the head here. This hydrophilic means it likes water, and a hydrophobic tail, right? Hydro, water, phobic, doesn't like water, tail. And so these molecules are all set up in this fashion because what surrounds a cell, both inside the cell and outside the cell, is water, right? Our bodies are made up of lots and lots of water. And so those hydrophilic heads, the heads of these molecules that like water, are pointing either to the extracellular space outside of the cell or the intracellular space, the inside of the cell, because that's all water. And those tails, those hydrophobic tails, face inside because they don't like water. And this makes up the membrane that surrounds the cell. Here's another picture. Again, you've got the phospholipid bilayer, the two layers, the hydrophilic heads that are pointing out either inside or outside of the cell, and the tails here that don't like water pointing towards each other. And you'll see that embedded in the membrane are these protein molecules that actually are what we call channels, and they allow substances to either enter or exit the cell. And we'll talk about them later, but you can see that they're embedded throughout this membrane. Now, neurons are similar to other cells, but they have a very unique shape. And all neurons have the following components that we'll talk about: dendrites, a soma or cell body, an axon, and a presynaptic terminal. So I'm actually going to show you, here's a picture of a neuron. Here are the dendrites, the soma, the axon, and the presynaptic terminal. Now what I want you to remember is that the purpose of these neurons is to send information. So imagine like neurons that start in your brain, and it's sending a message from your brain saying, I want you to type on the keyboard. So we've got to send a message from our brain down our spinal cord, down your arm, to your fingers, till you finally start typing on the keyboard. So that's a lot of neurons. Remember, there's a lot of neurons here all in a row. And they are trying to send a message, right? There's a neuron here sending a message to this neuron, and imagine this neuron sending a message to another neuron, all the way until we get from our brain to our fingers. All right, so we're going to start with dendrites as the first part of the specialized part of the neuron. These are branching fibers that are responsible for bringing information into the neuron. So they receive information and bring it into the cell body. So remember, there are all sorts of neurons all over here sending information. Let me just see if I can get a pen. Sending information to this neuron. That's supposed to be an arrow. Okay, so there are neurons here, and they're all sending a message, like maybe the message to say, hey, we want to move our fingers and type on the keyboard. So these dendrites are here to sort of imagine them like branches of a tree or like fingers on a hand reaching out to catch the information that these other neurons are sending. Okay, so that's the dendrites. The information then goes to the soma, or the cell body, which is right here, and this is the part that houses all those typical parts of a neuron, the nucleus, the mitochondria, the ribosomes, etc. So the soma, or the cell body, contains the nucleus, the mitochondria, all the other structure found in all the typical animal cells. But then we have another very specialized structure here, this in yellow, is called the axon. And this is a long tube that allows a neuron to send information down a particularly long distance, right? It's like they stretched out this animal cell and made this axon so that it can send its message, right? If you've got to send a message from your brain to your fingertips, think about how long of a space that is, right? Is that 2 feet, 3 feet? Now, the neurons aren't that long. There are individual neurons that are all communicating with each other. But if you can have your neuron be 1 inch long or 2 inches long, boy, that's great. That could help get the message to your fingertips even more quickly. And so the axon is this thin tube that transmits information. So a thin fiber that sends information from the cell body to the presynaptic terminal. I'm going to skip right down here to the presynaptic terminal. Oops, sorry. The presynaptic terminal is what we call the end of the cell. Basically, it's just the end of the cell. Why do we call it the presynaptic terminal? Sounds like a really fancy word instead of just saying end of the cell. But that's what it is. It's the end. Okay? Excuse me. So the presynaptic terminal is the end point of the axon. And we'll find out that that end point actually releases chemicals. I'm going to come back to myelin sheath in a minute. So here we go. Here's another just different view of a neuron. Here are these dendrites, these red dendrites that are reaching out, trying to get information from other nearby neurons. And you can see in kind of like this peachy color, these are other neurons that are nearby trying to send information to this cell. And so the dendrites are out there kind of reaching out to get information. The information is then sent to the soma, or the cell body, which is right here, that has all of the parts of a cell, the nucleus, the ribosomes, the mitochondria, etc. Information is then sent down the tube, that's called the axon, to the end of the cell, called the presynaptic terminal. Okay? Now, the axon, and we will talk about this another day, is covered by, or many axons are covered by what we call a myelin sheath. And this is an insulating material that covers the axon in many neurons, and it actually speeds up communication along the axon. And we'll talk about how it does that. But the myelin is this purple stuff that's covering up the axon, and it helps this message get sent more quickly, which is important because you want to send messages quickly. You want your nervous system to work quickly, right? You need to be able to do what you want in as quick of a time as possible. And so the myelin sheath allows even quicker transmission. In this picture, it's this blue stuff. That would be the myelin sheath, this fatty covering around the axon. Here's a real neuron looking at it through a microscope with the soma, the dendrites. Here's the axon and the presynaptic terminal, the end of the cell. Another picture. But you can see in real life it's a lot more complicated, right? Because we said there's 100 billion neurons all smushed together in your brain. And so it's hard to see, right, what we're talking about, this very simple dendrites, soma, axon, terminal, like it's straight out in a line. These are all on top of each other, all twisted around each other. And you can see it's very complicated when you're looking at a neuron or trying to look at a neuron in an actual brain. So neurons vary in their size, their shape, and their function. Their size, they can be virtually non-existent, microscopic, to about a meter in length. There is a neuron that goes from your spinal cord to your feet. So you can look at how long your leg is, and that's basically how long that neuron is. And then the shape can be very different. How much branching, how many dendrites determines how much it communicates with other neurons, right? The more branching, the more dendrites, the more communication. And so the function of the cell is closely related to the shape of the neuron. There are certain cells called Purkinje cells. They're in the cerebellum. Again, we'll talk about this as we move through the class. And they branch extremely widely. They have a lot of branching because they're responsible for motor coordination. And if you think about your motor coordination, you need a lot of things to be communicating. Your visual input, your balance, your motor muscles, all sorts of information has to be kind of integrated in order for you to have smooth motor coordination. So these Purkinje cells branch very widely because they have to take in a lot of information in order to do their job properly. All right. Three main types of neurons, although there are many different kinds. We'll talk about a few here, called motor neurons. Motor neurons are very large with long axons, and they end up attaching to a muscle. So a motor neuron is sort of the end of a pathway of neurons that ends up communicating with a muscle. It has its soma, or its cell body, in the spinal cord, and it receives a message from other neurons, and then it conducts those impulses. It sends its message along its axon until it gets to a muscle or a gland. And that's actually the picture that we were looking at, a motor neuron. Here it is with the dendrites reaching out to receive information from other neurons, the cell body, the axon, and then you can see that the neuron ends, the presynaptic terminal, is on a muscle fiber. So this might be a muscle telling you to type on your keyboard or kick your leg or walk or something like that. There are also sensory neurons that we'll talk about. These have different shapes to respond to environmental stimuli. So these are neurons that are specially designed to receive sensory information. So we have sensory neurons in our eyes that are able to detect light, therefore we're able to see. We have sensory neurons in our ears, so they are specially designed to be able to pick up sound, and therefore we can hear. And we have sensory neurons in our skin so that we can feel things. All right, and so that's an example here of a sensory neuron. So sensory neurons here start in the skin. In this case, this is a touch neuron. And once someone touches your skin, maybe this feels pressure, or it feels heat, or it feels something, it sends a message down the axon, or you can kind of imagine, for our purposes, there's a soma, and then goes down the axon, and then to the presynaptic terminal, or the ends of other cells. And this is sending a message this way from the outside world into your brain, right? Into the spinal cord and up to your brain, saying, ooh, I just felt something. Again, the soma, you could imagine it closer here. There's all different shapes. And then interneurons, these are tiny neurons that have very small axons, and they're between neurons. So they receive input from one neuron and send it to another neuron. They're the in-between kind of messengers. And in this way, right, of thinking about sending information from your brain out towards your muscles, or from the world like light or sound or touch information into your brain, we have these words, afferent, efferent, and intrinsic. So afferent axons refer to bringing information into a structure. Efferent refers to carrying information away from a structure. So in this case, cell A is sending information, right, to cell B. So this is efferent. It's sending information away. And this cell here is gaining information, bringing information in, or afferent. And those interneurons or intrinsic neurons are those that are between two neurons, and they're basically just sending or kind of sending information and passing the message along. Let's talk about glia cells for a minute. There are many types of glia cells. Each do sort of their own thing. We've got astrocytes. Those are star-shaped glia cells that remove waste, including dead neurons. So in order for your neurons to function correctly, they need all the extracellular fluid, the fluid that surrounds the neurons, to be clean, be at have specific levels of certain ions, etc. And so the astrocytes remove any kind of waste. That's their job, and get rid of it to kind of keep the neurons surrounded by clean fluid so they can work properly. Microglia also remove waste and other microorganisms that might get into that fluid around the neurons. Radial glia, those are glia that guide the migration of neurons during embryonic development. So as the brain is developing, these radial glia, they almost act like ladders, and you can see there's these amazing microscope images of the neurons sort of climbing along those radial glia to get to wherever they're supposed to be in the brain. Oops, sorry. Now, we talked about the myelin sheath that covers the axons and that allows messages to be sent more quickly down the axon. And myelination is actually accomplished by glia cells. And there are two different types of glia cells. The oligodendrocytes are a type of glia cell. They're located in your central nervous system. CNS is central nervous system, which means your brain and spinal cord. We'll be talking about this definition in an upcoming lecture. Located in the central nervous system, so the brain or spinal cord, and they produce the myelin for axons in your brain and spinal cord. One oligodendrocyte myelinates many axons. And so here's a picture. Here's an oligodendrocyte. This is a glia cell. And you can see it almost has many arms. And these arms can reach out and wrap around an axon and create myelin. And you can see here's another arm reaching out and creating myelin on another axon. So in this way, one oligodendrocyte can myelinate many axons and many different neurons. So one oligodendrocyte myelinates many axons. Now, when the axon is damaged, the oligodendrocyte actually produces scar tissue and prevents the healing of the axon. So if this axon is broken, and we're talking about in the brain or spinal cord. So let's imagine that somebody has breaks their back, and they actually break through this neuron. So information can't be sent, right? That's a problem, and that's what causes paralysis and things like that. The oligodendrocytes are very unhelpful in this situation. They actually imagine there's a break right here. Okay, the oligodendrocytes actually produce scar tissue. And so even though these axons are trying to connect again, they can't because of the scar tissue made by the oligodendrocytes, which is why damage in your central nervous system, brain or spinal cord, is pretty much permanent. Now this is in contrast to the Schwann cells. Those are located in our peripheral nervous system. There are also glia cells that make myelin, in our peripheral nervous system, and those are your nerves anywhere outside of your brain and spinal cord. So you have nerves in your fingers and your toes and your legs, right? If you feel something, you've got a nerve there. Now, here's a Schwann cell. The Schwann cell, imagine the Schwann cell is a big cell, and then when it sees an axon, it wraps its entire body around the Schwann cell. So each segment of myelin is an entire Schwann cell. So one Schwann cell is creates or can only myelinate one axon, different than the oligodendrocytes. One Schwann cell myelinates one axon. And then when an axon is damaged in the peripheral nervous system, so not in the brain or spinal cord, the Schwann cell actually creates a tube that allows the axon to heal. So if there was a break in a neuron, and you've all done this, if you've cut your finger, you've cut through neurons, it doesn't mean you permanently can't feel something in your finger. A Schwann cell will actually kind of wrap around the two parts of the broken neuron, broken axon, make a tunnel basically that helps the two parts of the axon grow back together. And so the Schwann cells are very helpful for regrowth of axons in the peripheral nervous system, whereas the oligodendrocytes are the opposite, right? They make like a scar here and don't allow the two parts of the axon to reconnect. And so this is actually where there's a lot of research, because if we could figure out how to make oligodendrocytes act more like Schwann cells, then damage in the central nervous system in the brain and spinal cord may not be permanent. Here's a picture of an oligodendrocyte myelinating these many axons, and here's a real microscopic picture of how that happens. Blood-brain barrier. There is something called the blood-brain barrier that surrounds the brain and blocks most chemicals from entering. The immune system destroys damaged or infected cells throughout the body, but because neurons in the brain generally don't regenerate, remember, once they're damaged because of those oligodendrocytes and other reasons, they don't grow back. The blood-brain barrier must block any incoming viruses, bacteria, or harmful material from entering the brain. And so this happens by endothelial cells of the walls of your capillaries joined tightly to form what we call this blood-brain barrier. We're going to watch a quick video on the blood-brain barrier. It may be a little technical at times, but just to get the idea of how this works.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the blood-brain barrier. The blood-brain barrier is a complex that surrounds most of the blood vessels in the brain. It acts as a barrier between the bloodstream and the extracellular space of the brain, allowing only certain substances like water, oxygen, and small lipid-soluble substances to easily cross from the blood into the brain. This prevents toxins, pathogens, and other potentially dangerous substances from crossing from the circulatory system into the brain. It is thought that the central components of the structure of the blood-brain barrier are the tight junctions of endothelial cells, the cells that make up the interior surface of blood vessels. In other blood vessels throughout the body, there are small spaces between these endothelial cells. Small blood-borne substances can pass through these spaces and into surrounding tissues. The endothelial cells that make up the blood-brain barrier, however, are fused tightly together to form tight junctions that restrict diffusion across the blood vessel lining. Glial cells called astrocytes also have projections called astrocytic end feet that extend to the walls of blood vessels that are part of the blood-brain barrier. Astrocytic end feet often completely surround blood vessels in the brain and are thought to play critical roles in the formation of the blood-brain barrier. For example, they seem to be involved with signaling that prompts endothelial cells to form the tight junctions necessary to create the blood-brain barrier.
So you can see that because of these endothelial cells of the capillary walls that are joined very tightly together to form the blood-brain barrier, they basically allow small uncharged molecules like oxygen and CO2 and some to pass through the blood-brain barrier. However, although we're trying to keep out things that would potentially harm the brain, some fat-soluble molecules like heroin, marijuana, nicotine are also able to cross the blood-brain barrier, and that therefore you get that those cognitive effects of these types of substances, which we'll talk about in this class. And so the brain needs to use active transport, so energy transport, to pump in to the brain glucose, amino acids, vitamins, and hormones. So instead of just passively having these things kind of flow in and out of your circulatory system and the rest of your body, because damage to the brain can be permanent, we have these tight junctions of the blood vessels that really only allow carbon dioxide and oxygen to flow through. Everything else has to be actively pumped into the brain. All right, and here's a picture of these tight junctions. So the endothelial cells are tightly basically touching each other and only allows oxygen and carbon dioxide to pass from the bloodstream into the brain. And these large molecules aren't able to get in, which hopefully protects the brain so you don't get viruses and bacteria and things like that. All right, thank you, everybody.